human immunity system is a constantly evolving network of cells. But how does it adapt to certain viruses and bacteria? Well, the answer lies in a small protein called an antibody. These tiny Y-shaped proteins participate in the fundamental recognition of viruses and are imperative to our overall adaptation to disease. Without them, we the human race would cease to exist. So how are these essential tiny proteins created? To understand this, we start by investigating the fundamental cells of the adaptive immune system. The first major component is something called a B cell. Like most of the cells that we'll be talking about today, they originate in the bone marrow. After creation, they directly travel to a lymph node or the spleen. The next prominent cell is the T cell. Like mentioned earlier, they also start in the bone marrow. However, the one differentiating factor is that instead of going directly to lymph nodes, they go to an organ called the thymus. While in the thymus, they go through a very harsh training process where only 1/4 of the T cells that entered survive. This intense training equips the T cells with very specific setups so they can carry out individualized roles. After training, T cells reside in lymph nodes and the spleen as well. Continuing on the path to our overall goal of antibody production, the adaptive immunity system must first be activated. There's two ways that this happens. The first uses a dendritic cell who collects samples at the battle site. After getting the new antigen, it gives the knowledge to new naive B cells in the lymph nodes. The second utilizes the actual lymph system, and through this, naive B cells are able to actually grab the antigen as it flows through areas such as the spleen and lymph nodes. The overall concept is that a naive B cell has its hands on a new antigen. So what happens next? Well, this is when something truly fascinating occurs. When a naive B cell encounters a new antigen, it initiates something called clonal expansion. Through this process, B cells and T cells start rapidly multiplying and cloning themselves in a somatic method. This allows the B and T cells to diversify their abilities. They break up into a number of subcategories. The first one is helper T cells. These T cells will help facilitate the overall process of making antibodies and will help the adaptive immune system work as a whole. The second are the killer T cells, the ones that will actually go to the bacterial infection site and fight the bacteria. Think of them as extra reinforcements for the innate immune system. The third subcategory is regulatory T cells. This cell has the responsibility of stopping the innate immune system if it recognizes its own cell as an enemy. This effectively stops us from tearing ourselves apart from the inside. The fourth are memory T cells, the staple of the adaptive immune system. These cells stay in the lymph nodes or go back to the bone marrow, so in case of a reinfection of the same virus, they are already prepped for the process and ability to kill that same cell. When our adaptive immune system encounters a new antigen, it normally takes a few days for it to create a process to kill it. By having memory T cells and memory B cells, we have instant protection that has already been set in place and trained. But how is that process even created? Well, that features our final subcategory, the plasma B cells, the ones responsible for creating actual antibodies. After grabbing an antigen, they start their training process. This instruction is a lot of trial and error and really just finding what antibody has the best connection to the antigen. Because they have to be so quick in their recombinations, plasma B cells will actually cut up chromosome 14 and only take the vital parts to make an antibody. You see, antibodies are composed of a leader segment, a variable region, a diversity segment, a joining segment, and a constant region. These are dispersed throughout the entire chromosome 14, meaning that if they were to use the regular protein synthesis method, the adaptive immune system would fail to find the right antibody. The small cutout region becomes the gene segment that is transcribed. Other than this, the protein synthesis process stays the same. The enzyme RNA polymerase binds to the gene segment and creates an mRNA. After this, the mRNA will flow into the cytoplasm and meet the ribosome. The ribosome will use it as a blueprint and use the mRNA sequence in groups of 3 bases called codons. The ribosome then attracts transfer RNAs carrying specific amino acids that bind together to create our proteins. After this, it starts something called the secondary structure, where it either forms an alpha helix or a beta pleated sheet. Then it moves on to the tertiary structure, which forms actual three-dimensional shapes. And it ends with the quaternary structure, where using multiple proteins creates a complex fold of polypeptide chains. Because of clonal expansion, millions of these tiny antibodies are created. So where do these millions of Y-shaped proteins go? Well, after formation, it directly shoots towards the bacterial infection site where it starts attacking the enemies. It does this in multiple ways, the first being antigen dysfunction. When a bacteria attacks, it uses its antigens to bury into a cell. So, when an antibody attaches to these antigens, it effectively removes its ability to attack our cells, allowing our macrophages and innate immune system to easily clean them up. A similar process is used in its second function, which is clumping, where the antibodies connect to each other, grouping the actual bacteria into small concentrated areas, allowing the innate immune system to kill efficiently without harm. In conclusion, while these antibodies may be the smallest protein in our immune system, they play an extremely important role for long term immunity and the future of our human bodies. Thank you.